Welcome to Biology at Priyajit, my dear viewers. Today we'll explore the molecular basis of DNA repair in prokaryotes, the essential mechanisms that maintain the genome integrity. We will understand how photoreactivation repair reverses UV-induced damage, how base and nucleotide excision repair remove and replace damaged DNA segments, and how the SOS repair system acts as an emergency response to serve DNA damage. Finally, we will decode the RecBCD model of repair, a crucial pathway involved in homologous recombination and double-strand break repair in bacteria. Get ready to uncover how cells protect their DNA at the molecular level. DNA repair mechanisms, bacteria to humans. Why are DNA repair mechanisms important? Mutations can damage genetic material. Too many mutations can be harmful or lethal. Therefore, organisms must have evolved multiple DNA repair systems, and these systems help maintain mutations at a tolerable level. DNA repair mechanisms in E. coli. The bacterium E. coli has 5 well-characterized DNA repair mechanisms. First one is the light-dependent repair or photoreactivation, excision repair, mismatch repair, post-replication repair, and error-prone repair system, that is SOS response. So DNA repair in mammals. Mammals possess almost all DNA repair systems found in E. coli. Exception is the photoreactivation process. Why? Because most mammalian cells do not receive visible light, therefore light dependent repair is not useful in mammals. Light dependent repair or photoreactivation. Light dependent repair, or also called as photoreactivation, is a DNA repair mechanism in bacteria. It is carried out by a light activated enzyme called DNA photolyase. Cause of DNA damage: exposure to ultraviolet UV light damages DNA. UV light causes formation of thymine dimers. Thymine dimers are formed due to covalent cross-linking between adjacent thymine bases. This distorts the normal structure of DNA. Role of DNA photolyase. DNA photolyase recognizes thymine dimers and they bind specifically to the damaged site in DNA. It uses energy from visible light to repair the damage. Requirement of light. We know that photolyates can bind to thymine dimers even in the dark. However, it cannot break the covalent bonds without the light energy. So it requires visible light, especially light in the blue region of the spectrum. The enzyme uses this energy to cleave the crosslinks. This is the image showing the cleavage of thymine dimer crosslinks by light activated photolyates. Step 1, this is the UV reaction. Thymine dimer is formation, then photolyase binds to the thymine dimer in DNA, then cleavage of crosslinks. Photolyase is activated by the absorption of the blue light and then release of the enzyme. So other dimers also repaired in this pathway. DNA photolyase can also repair cytosine dimer, cytosine and thymine dimer, etc. Experimental significance: when UV light is used to induce mutations in bacteria, the cells are grown in the dark. Reason: to prevent photoreactivation, and this helps maximize mutation frequency. Excision repair. Excision repair is a DNA repair mechanism that removes damaged DNA segments and replaces them with correct nucleotides. Basic steps of excision repair. Excision repair involves three main steps, that is recognition and removal. A DNA repair endonuclease or enzyme complex recognizes the damaged base, binds to the site, excises or removes the damaged portion. Gap filling or DNA polymerase. It uses undamaged complementary strand as a template, synthesizes new DNA to fill up the gap. Sealing. DNA ligase. It seals the DNA and restores DNA continuity. So there are two basic major types of excision repair. First one is the base excision repair or BER, and second one is the nucleotide excision repair or NER. Both occur in the dark, operate in both E. coli and humans, and follow similar general mechanisms. Base excision repair. It removes abnormal bases and chemically modified bases. Examples: deaminated bases or oxidized bases, etc. Steps of base excision repair recognition. It is initiated by enzymes called DNA glycosylases. Each glycosylase recognizes a specific altered base. Removal of abnormal base. Glycosylase cleaves the glycosidic bond. It separates base from 2-deoxyribose. It creates epi site or apurinic site or apyrimidinic site. Third step is the removal of sugar phosphate. Epi-endonuclease recognize the epi site along with phosphodiesterase enzyme. It removes sugar phosphate residue. Gap filling DNA polymerase, it inserts correct nucleotide. Sealing DNA ligase seals the nick. See, this is the image showing the mechanism of base excision repair. Nucleotide excision repair, it removes thymine dimers, bulky lesions, bases with large side groups. Mechanisms in E. coli organism. E. coli mechanism enzymes involved. Excision nuclease called XC nuclease requires gene products like UVR, UVRB, UVRA and UVRC. Stepwise mechanism in E. coli, a trimeric protein, 2 UVRA and 1 UVRB. It recognizes DNA damage, binds to the damaged site and uses ATP to bend DNA. UVRA dimer is released, then UVRC binds to the UVRB DNA complex. UVRC makes cuts, fourth or fifth phosphorylation bond on 3' side and 8th phosphorylation bond on 5' side. UVRD, that is DNA helicase 2, it removes the excised 12 nucleotide fragment or dodecamer. Then DNA polymerase 1 fills the gap and DNA ligase seals the nick. So this is the image showing the process of nucleotide excision repair. Nucleotide excision repair in human, which is similar to E. coli, but it is more complex. It requires about 4 times more proteins. Exonuclease contains 15 polynucleotides. So key protein, XPA protein, which recognizes damaged DNA, recruits other excision proteins and which are associated with xeroderma pigmentosum. Difference, this is the table showing the differences of nucleotide excision repair in human from E. coli steps. So final steps in human, DNA polymerase delta or epsilon fills the gap and DNA ligase seals the nick. So this is the image showing the process of DNA repair. Other DNA repair mechanisms include living cells. We know that possess an army of DNA repair enzymes that constantly scan DNA for damage, such as thymine dimers caused by UV light, base mismatches, chemical modifications, strand breaks, bulky DNA lesions, etc. Recent research has also discovered new DNA polymerase that play special roles in DNA repair. Mismatch repair or MMR, it corrects mismatched bases left after DNA replication. It acts as a backup to DNA polymerase proofreading. Example of mismatch, T paired with G, that is TG mismatch. Both bases are normal, so the system must identify which strand is correct and which strand contains the error. How does bacteria distinguish the strands? In case of E. coli, we know the sequence GATC gets methylated and after replication, parent strand is methylated and newly synthesized strand become unmethylated. So the temporary difference helps the repair system identify the wrong base. Proteins involved in mismatch repair or E. coli, it requires genes like mut-H, mut-L, mut-S, mut-U or UVRD, etc. Stepwise process. Mut-S, it recognizes and binds mismatch. Mut-L and mut-H join. Mut-H then cuts the unmethylated strand at nearby GATC site. Excision, if cut is 5' to mismatch, then 5' to 3' exonuclease acts. If cut is 3' to mismatch, then 3' to 5' exonuclease acts. DNA polymerase 3 then fills the large gap up to 1000 base pair. DNA ligase, it seals the nick. And mismatch repair in eukaryotes, we know that homologs of mut-S and mut-L exist in fungi, plants, and mammals. Therefore, mismatch repair is nearly universal in all organisms. So this is the basic process where the mismatch repair system is shown. Post-replication repair or recombination repair. When does it occur? When DNA polymerase 3 encounters a thymine dimer, polymerase then skips over lesion and it leaves a gap opposite damaged site. So this is the step-by-step process which is controlled by RecA protein. RecA protein promotes homologous recombination. This gap is filled using the sister DNA strand as template, and DNA polymerase fills remaining gap. DNA ligase seals the nick, then thymine dimer remains unless removed by nucleotide excision repair system. SOS response or error prone repair system, when it is activated when DNA damage is severe like a heavy UV exposure. What happens? Cell activates emergency system. Many repair genes are expressed at that time. Key regulatory proteins involve LEX-A and RecA. In normal condition, LEX-A represses SOS genes, but after DNA damage, RecA binds to single-stranded DNA. RecA activates itself. It stimulates LEX-A self-cleavage, and SOS genes are expressed. Error-prone polymerase, DNA polymerase 5, then encoded by UC and UD genes. It replicates across damaged DNA. In inaccuracy causes increases mutation rate. So SOS response is risky but helps in survival. Recombination, we know recombination is a process where DNA segments break and rejoin. Types include homologous recombination, site-specific recombination, and transposition. Homologous recombination is the exchange between DNA segments that are similar or identical. It occurs during meiosis 1 crossing over and occurs occasionally in mitosis. Holliday model in 1964 was proposed by Robin Holliday. That includes step-by-step process, alignment of homologous chromatids, single-strand breaks or nicks, strand invasion, formation of Holliday junction, then branch migration and resolution. So this is the image showing the step-by-step process of homologous recombination. So what are the key concepts? Holliday junctions makes a cross-shaped structure. Branch migration causes movement of crossover point. Heteroduplex DNA region with mismatched base, and resolution is performed by cutting and rejoining DNA strands. Modern models of recombination, Michelson-Rading model 1975 initiated by single nick, and double-strand break model was proposed by Jack Szostak and Franklin Stahl, most widely accepted model. So these are the double-strand break model steps. Double-strand break occurs, DNA ends are degraded, that is single-stranded region, then strand invasion forms D-loop, DNA gap repair synthesis occurs, two Holliday junctions forms. So resolution, recombinant or non-recombinant chromosomes. So in conclusion, DNA repair systems are vital for maintaining genomic instability in prokaryotes. Photoreactivation repair directly reverses UV-induced DNA damage, while base excision and nucleotide excision repair precisely remove and replace the damaged DNA segments. The SOS repair system repairs an emergency response to extensive DNA damage, and the RecBCD pathway plays a central role in double-strand break repair through homologous recombination. Together, these mechanisms ensure DNA integrity and cellular survival, highlighting the remarkable molecular strategies that protect life at its core. Thank you.